Welcome everyone to Chapter 8, Patterns of Inheritance. In our last lecture, we spent a lot of time learning about how eukaryotic organisms produce the cells known as gametes, which are the ones that serve as the basis for sexual reproduction. And now in this chapter, we're going to learn all about how when those gametes come together to form a new organism, they bring with them a set of genetic information that manifests in the offspring as their physical characteristics and traits. And I want to let you know ahead of time that this is one of the longer lectures of the semester because it's pretty detailed. So you probably want to be prepared to break this one up into pieces in order to get through it. Now, the photo that you see on the title slide here is of pea plants. In those green pods that you see hanging, that's where you can find peas, which are the offspring, the babies of the pea plant. And why do I have a photo of a pea plant here? That will become clear shortly. But first, let's define what we're talking about when we say patterns of inheritance, the title of this chapter. Inheritance is the genetic passage of traits from parents to offspring. The DNA in our cells is what brings about many of our characteristics. Not all of them, of course. Many others of our characteristics are things that we've learned or acquired or been conditioned into throughout our lives. But that's not what we're talking about when we refer to biological inheritance. In biology, inheritance refers to traits encoded in DNA that are transmitted from parent to offspring. And this is something that we can plainly see when we just look around us. We all know that children tend to look like their parents. Sometimes they look more like one parent than the other, but you can often pick out certain traits and say, oh, she has her mother's eyes, or she has her father's smile. And yes, I did use photos of celebrities in this lecture because they may be faces that are more known to us. So these are the actors Uma Thurman and Ethan Hawke and their daughter Maya Hawke, who is also an actor. And here's another example. This is tennis star Serena Williams and her husband and their daughter. And while you can't really see their faces, you can see that their daughter's skin tone is a blend of both of her parents. It's somewhere in the middle of the two shades. And examples of characteristics like this one, like the skin tone, led to the development of what used to be called the blending inheritance hypothesis. The blending inheritance hypothesis is a now obsolete idea about how inheritance worked, which was that for each trait, offspring inherit an average of their two parents. So if one parent has darker skin and the other has lighter skin, their children will have a skin tone that's in the middle. One parent's tall and one parent is short, their children will be somewhere in the middle. And this hypothesis was proposed not just on the basis of looking at human characteristics, but characteristics in other organisms as well. For example, in snapdragon flowers, when a plant that has red flowers pollinates a separate plant that has white flowers, their offspring are pink, halfway between red and white. So these are the types of observations that led biologists of an earlier era to propose the concept of blending inheritance. However, there was a problem with blending inheritance, and there's a reason why this hypothesis is now obsolete, because is it always the case that offspring inherit the average of their parents' traits? We don't need to look very far to find some glaring exceptions to this idea of blending inheritance. Take this scenario, for example. This is a photo of a Labrador mother and her litter of puppies, and the mother has a black coat, and then the father is the dog whose face is highlighted in the smaller box at the top. The father also has a black coat. But as you can see, their litter of puppies is a mix of black, yellow, and chocolate brown labs. So if blending inheritance was true, this wouldn't be possible. So how is it possible? And no, although it is tempting, the correct answer is not that this is not the real father. These dogs are both the real parents of this litter of puppies. So naturally, we have to scrap the blending inheritance hypothesis because it simply does not hold up in instances like this. But if blending inheritance isn't true, what is? What is the real mechanism by which offspring inherit genetic characteristics from their parents. So enter this guy, Gregor Mendel, a German biologist and monk who is credited with discovering the true mechanisms of inheritance that overturned this blending inheritance hypothesis. So in the mid-1800s, Mendel performed extensive studies on pea plants, and he wrote about his findings in a manuscript that he titled Experiments in Plant Hybridization. And while the importance of his discoveries was not really recognized until many years after he died, he is considered to be the founder of modern genetic theory. So in this lecture, we are going to take a look at exactly what Mendel did in his experiments and how that led him to discover how inheritance works. But first, why pea plants? Why did Mendel choose pea plants as his subjects to study? So peas have a lot of advantages, including the fact that they are small in size, so you can cultivate a large sample size without needing much space. They also grow and reproduce quickly. It only takes about 60 days for a pea plant to reach maturity. Pea plants also have many different easily visible traits that can be studied, and they had been studied extensively by other researchers before Mendel, so he had something to go on. And lastly, pea plants can be fertilized in a controlled manner. And that's what you see happening in this illustration right here. This drawing is showing Mendel manually pollinating a pea flower. So what he's doing here is the same thing that pollinator insects like bees would do in the wild. So he has this little paintbrush-like tool, and he has picked up pollen from one of the flowers, and he's transferring it to the flower of another plant. And in this way, Mendel was able to control which plants were breeding together. He was able to decide and control which plants would be the parents, and he made those choices based on what he saw in the parents' traits. And then he observed how their offspring looked after interbreeding them. And it's important to note that when we're talking about pea plants, they are not quite like humans in terms of how they reproduce. So pea plants have both male and female reproductive parts within the same plant. So on the left, you can see what a pea flower looks like on the plant. And then on the right, this is a very close-up image of a flower that's been sliced down the middle. So within the flower, there's a part called the anther, which is shown in the smaller white box, and that's where the pollen is stored. And the pollen is what contains the sperm, which is the male gamete. And then now I'm highlighting in the white rectangle, this is the ovary. And you can see in the ovary, it contains these little round cells that are called ovules. And the ovules are the equivalent of eggs in humans. There's the female gamete. So as you can see, both the male and the female parts are contained within the same plant. So this means it's possible for pea plants to mate with themselves. And when they do so, this is called self-pollination. The pollen in the flower enters the ovary and fertilizes the ovules within that same flower. So in some experiments, Mendel would allow his pea plants to self-pollinate, but on other occasions, he would deliberately cross-pollinate specific parents with each other. So he'd collect pollen from one and then transfer it to the ovary of another. And to guarantee that the plant receiving the pollen didn't inadvertently self-pollinate itself, he would remove the anthers from that plant. So this is how he would control which parents were breeding with which. In all of his experiments, though, Mendel always started with true breeding pea plants. A true breeding organism is one that always produces offspring that look like the parent. So to guarantee this, Mendel would allow one variety of pea to self-pollinate for multiple generations to confirm that it was true breeding, meaning to confirm that it always produced offspring that looked like itself, before he would use it in his experiments. Okay, so now we know a little bit about Mendel's subjects of study, the pea plants. Now let's see what he did in his first experiment. One of the characteristics that Mendel looked at in his pea plants was the texture of the seeds. In other words, the texture of the peas themselves. The peas are the seeds of the pea plant. So for this characteristic, there are two variations. Some peas have a smooth texture, which is what you see on the left, and some peas have a wrinkled texture, which you see on the right. So what Mendel did was he found a variety that was true breeding for smooth, round seeds. And then he took another variety that was true breeding for wrinkled seeds, and he cross-pollinated them. And what he found is that 100% of the offspring of this cross were smooth and round. Not one of them was wrinkled. They all looked exactly like the first parent and nothing like the second parent. So already at this point, blending inheritance has to be thrown out. Because if blending inheritance were true, then you would have seeds that were intermediate between wrinkly and smooth. Now, this first generation he referred to as the F1 generation. But Mendel did not stop at the F1 generation. He went a generation further. So he planted the seeds from the F1 generation. He allowed them to grow to maturity and self-fertilize. So in other words, the pollen and the ovules from the same plant combined. And when that happened, in the following generation, he saw the wrinkled texture reappear in a portion of the offspring. So this next generation, these are known as the F2 generation. They're 2 steps away from the original parents. And within this generation, Mendel found that 5,474 of the offspring seeds were smooth and 1,850 were wrinkled. And yes, Mendel really did count thousands of peas. Evidently, he had a lot of time on his hands. Now, if you look at these numbers, it may not be immediately obvious, but they have a very specific ratio to each other. And so in this first checkpoint, I want you to calculate what that ratio is. What is the ratio of round peas to wrinkled peas resulting from this first experiment? So to find this, you divide the first number by the second number, and you can round to the nearest whole number when you're coming up with your answer. So Mendel's first series of experiments didn't stop there. He ended up doing that exact same series of experiments with other characteristics as well, such as the color of the peas. So as with texture, there are 2 variations on color. There's yellow seeds and green seeds. So Mendel took pea plants that were true breeding for yellow seeds, and he crossed them with plants that were true breeding for green seeds. And once again, what he found was that the first generation had offspring that all looked like one parent, but not the other. They were all yellow. But then when he planted those yellow seeds and allowed them to grow up and self-pollinate, the next generation showed a reappearance of the green trait. Specifically, 6,022 of the F2 generation were yellow and 2,001 were green. And with these numbers, you can probably more easily tell just by looking at them what the ratio is. If you divide the first number by the second number and round it to the nearest whole number, it should be the exact same ratio that you saw with the characteristic of texture. So now we have a pattern starting to form, and there must be something going on here. But Mendel didn't stop there. He repeated this series of experiments with 7 different characteristics of pea plants, including the 2 that we just talked about, along with flower color, pod shape, pod color, flower position, and stem height. And in each case, there were 2 different variations of the characteristic being studied. He would cross 2 true breeding plants and find that all of the offspring looked like one parent. And then in the next generation, the other trait came back in the exact same ratio. So, how did Mendel explain this pattern that he saw over and over again? Well, he proposed that pea plants inherit 2 separate quote-unquote units, which is what he called them. Today, we would call them genes. One comes from the male parent and one comes from the female parent. And that when the parents produce their gametes, the 2 units that they possess are segregated, which is the term that he used, into separate cells. Now, we learned about this process very recently, actually. In our last chapter, we talked about the process that cells use to produce their gametes, and how their genes are separated into distinct cells through this process. So, refresh your memory from chapter 7. Tell me what that process for creating the gametes is called in this checkpoint. So, getting back to Mendel's explanation, this was only part of how he explained what he saw. He also proposed that for a given characteristic, one of the variations is dominant and the other is recessive, meaning that if an organism has one dominant unit paired alongside one recessive unit, only the dominant variation gets expressed. And these hypotheses that Mendel proposed turned out to be true, and over time they have become formalized under two names. The first explanation is now known as the law of segregation, which basically says when gametes are created, the parent's pair of genes gets separated, and only one ends up in each gamete. And the second set of explanations is now known as the law of dominance. Basically, one variation of a gene is dominant and overrides the other variation when they're paired up together. Now again, Mendel used the word units to describe these things that were being inherited, but we want to superimpose our modern terminology and understanding here. So today we use the word genes, which are sequences of DNA that encode a specific structure or function. A trait is a variation in the physical appearance of an inheritable characteristic. And then one more term to learn. Alleles are different variations of a gene that encode different traits. Now, in just a moment, I'm going to use these terms in the context of an example that will allow us to understand them better. But before we get there, let's very quickly review once again what we know about the content of genes, meaning what we know about DNA. Genes are made of DNA. So how is that DNA able to carry information about traits? Well, we've learned that DNA is made of repeating monomers called nucleotides. And those nucleotides themselves contain three internal parts. They've got a sugar, a phosphate group, and a nitrogenous base all linked together. And we also learned that these nucleotides are linked together in extremely long chains, and that each chain pairs with a second chain to create a double-stranded structure that represents your DNA. And what we haven't yet learned is that one of the three components of these nucleotides found within DNA, one of these three components can take different forms. And that component is the nitrogenous base, the one that I've just highlighted in the red box. The phosphate group is shown in blue, the sugar is shown in green, and those two are always, always, always the same. They look the same in every nucleotide found in the strand of DNA. But the nitrogenous base can vary. Specifically, it can vary between four different things, four different molecular structures. And you absolutely don't have to know anything about what these molecular structures are. I'm just showing the molecules here for your information, but you should know the names of the four different types. The first one is called thymine. The second one is called cytosine. There's also adenine, and then finally guanine. And it's common for these to be abbreviated as just their first letter. So thymine is abbreviated T, cytosine is C, adenine is A, and guanine as G. So when you have your double-stranded DNA, each strand will have a series of different nitrogenous bases. And to make it more clear in this image, here are those nitrogenous bases that have been hypothetically colorized. And here are the one-letter abbreviations placed on them. And if you take a second to look at this, you may notice that the order is not completely random between one strand and the next. So wherever you see a T, it's linked to an A in the opposite strand. And wherever you see a C, it's linked to a G in the opposite strand. And these are the linkages that hold the two strands together. Thymines are attracted to adenines, and cytosines are attracted to guanines. But as far as the order of the nucleotides within within a strand goes, not between strands, but within a single strand. It is the unique order of these letters that brings about different variations of a characteristic. These nitrogenous bases are quite literally like a four-letter language that spells out to the cell how to make things and how to do things. So let's take all this information and put it into the context of a specific example now. So humans have a gene in their DNA known as TAS2R8. And this gene is located on chromosome 12 on the P arm, which is the petite arm, the small arm, and position 13 of the petite arm. This segment of DNA determines whether a person is able to taste a chemical called phenylthiocarbamide, also known as PTC. PTC is a bitter tasting chemical that's found naturally in vegetables like broccoli and cabbage and Brussels sprouts. But not everyone can taste it, and the ability to taste it is genetically determined. Some people have the trait of, yes, they can taste PTC, while other people have the trait of, no, they can't taste PTC. And the difference comes down to what's written in the nucleotides that make up your DNA for that gene. And remember, different variants of a gene are called alleles. And these are completely hypothetical, but the sequence of nitrogenous bases for the allele that encodes the ability to taste PTC might read the way that you see on the left. And then the sequence of nitrogenous bases for not being able to taste PTC might read the way that you see on the right. And of course, I'm showing just one string of letters, and we know that DNA contains two strands, but this is the conventional way that scientists show the contents of a gene. They just show one strand. So you can see that between these two alleles, the letters are not the same. And this difference in the letters is what accounts for the difference in being able to taste PTC or not being able to taste PTC. But there's another fold to this, and that is the matter of which trait is dominant and which one is recessive. So in the case of PTC, being able to taste it is dominant, while not being able to taste it is recessive. And we know that Mendel saw this with his law of dominance, but let's put forth some formal definitions. A dominant allele is a variant of a gene that overrides the expression of other alleles when they are paired together. A recessive allele is one whose expression is masked by another allele when they're paired together. And when we're writing out dominant and recessive alleles, we often use a single letter abbreviation to represent them, just for the sake of convenience. So the dominant allele is represented by a capital letter. So for example, in the case of PTC tasting, we might choose capital T to represent the dominant allele. And then the recessive allele is represented by a lowercase letter. So in this case, lowercase t. And notice how these are the same letter. We didn't choose two different letters. We didn't choose, for instance, T for dominant and S for recessive. They're both T, just one of them is capital and one of them is lowercase. And any organism that is diploid is going to have two alleles for every characteristic. Because remember, our chromosomes come in pairs, and the genes that are found on these pairs of chromosomes, they're the same genes even if they represent different alleles. So the combination of alleles that you have on your pair of chromosomes is called a genotype. Genotype is the term that's used to refer to the alleles that an organism has for a given gene. For example, if you have the dominant allele on both of your chromosomes, this is your genotype. The counterpart to genotype is phenotype. Phenotype is the observed trait that is expressed by an organism. So if your genotype is that you have the dominant allele on both chromosomes, then your phenotype is that you can taste PTC. You can taste this bitter chemical that is present in many vegetables. That is the physical manifestation of what is found in your genes. So think phenotype, physical. Of course, this is only one possible genotype for this trait because there are two different alleles and every person has two chromosomes. And so therefore, there are four different genotypes that a person could have for this characteristics. You could have the dominant allele on both of your chromosomes, which is what we see here, but you could also have the dominant allele on just one of your chromosomes and then the recessive allele on the other. So let's say your mom passed down the dominant allele and then your dad passed down the recessive allele. Your genotype would then have one of each, but your phenotype is still that you can taste PTC because the dominant allele overrides the recessive allele. It's almost as if the recessive allele wasn't there at all. Likewise, your dad could have passed down the dominant one and your mom could have passed down the recessive one, which also would give you one of each. And again, this would mean that your phenotype was having the ability to taste PTC. And the last possibility is that both of your parents passed down the recessive allele, which would give you 2 copies of it. And in this case, you would not be able to taste PTC. There's nothing in your genotype to override the recessive alleles, so the recessive alleles get expressed instead of the dominant ones. Now, there are some terms that we use to describe these different genotypes. The 2 genotypes in the middle, the ones that have a combination of one dominant and one recessive allele, these are described as heterozygous. Heterozygous refers to a genotype that contains different alleles for a given gene. The prefix hetero means different or opposite. So that's what it means in this context too. 2 chromosomes with different alleles. And then the other 2 genotypes are described as homozygous. Homozygous means the genotype has the same allele on both chromosomes. So the prefix homo means the same, and there are 2 ways of being homozygous. To be homozygous dominant means that you have 2 copies of the dominant allele, and then to be homozygous recessive means you have 2 copies of the recessive allele. So now that we know all of this, let's get back to talking about Mendel's experiments. So you'll remember that one of the characteristics that Mendel studied was the texture of the seeds in the pea plants. So for this characteristic, we now know that smooth texture is dominant and wrinkled texture is recessive. And we're going to use the letter A to represent these. So capital A refers to the smooth texture allele, and lowercase a refers to the wrinkled texture allele. In this checkpoint, using those letters, capital A and lowercase a, I'd like you to list the different possible genotypes that any given pea plant could have for seed texture. And also, tell me what phenotype is produced by each of these possible genotypes. So now that you've determined the different genotypes that are possible for pea plants, let's look back at the parent plants that Mendel used and figure out what their genotypes were. We know that the plants that he used for his parent generation were true breeding, meaning that he had observed them repeatedly self-pollinating and their offspring always looked like them. So from this information, we can assume that their genotypes were homozygous. Specifically, the smooth parent must have had the genotype capital A, and the wrinkled parent must have had the genotype lowercase a. Now, when these parents created their gametes, only one of their 2 alleles will end up in each gamete. The chromosomes will get separated. So one goes into one gamete, the second one goes into the other gamete. And this is what Mendel meant in his law of segregation. But to really drive the point home, let's take a look at how the gametes would actually be created in the smooth pea plant parent. So this graphic is going to show the reproduction of a germ cell, which you may remember is the special type of diploid cell that gives rise to the gametes. So in the smooth pea plant parent, given that it is homozygous dominant, as 2 copies of the capital A dominant gene, the cell will go through its cycle and it will duplicate those genes when it copies the chromosomes during S phase. And then during meiosis 1, the chromosomes will be separated into 2 cells. And then finally, during meiosis 2, each one of those 2 cells splits again, and you end up with 4 haploid cells. And all 4 of them contain a single copy of the capital A allele. And because, remember, the pea plant has both male and female gametes within the same flower structure, this means that all of the ovules and also all of the pollen contain the capital A allele. So it is inevitable that this parent passes down the dominant allele to all of its offspring. And then when it comes to the other parent, the one with the wrinkled phenotype, this parent starts out with a pair of chromosomes that both have the little a allele, the recessive allele, and this allele will be doubled when the cell goes through S phase, and then the chromosomes get separated in meiosis 1, and then they get pulled apart again in meiosis 2. So consequently, all of the gametes in the wrinkled pea plant contain the lowercase a recessive allele. And again, this means that for the second parent, every single one of the female gametes in the ovary, and also all of the male gametes in the anther, contains the recessive allele. So it's inevitable that this parent passes down the recessive allele. And remember, Mendel crossed one of each of these plants with each other. So the male parent was true breeding for the dominant allele, and the female parent was true breeding for the recessive allele. So therefore, the male parent delivered a dominant allele in its pollen, and the female parent brought recessive allele in its ovule, and the fertilized embryos ended up with one of each. And this is the F1 generation. So if every member of the F1 generation has the genotype capital A, lowercase a, then this means that the phenotype, the express trait of all of those F1Ps must be smooth because the dominant allele overrides the recessive one. So this makes sense. So now let's take a pit stop for a checkpoint. Would these Ps, the members of the F1 generation, would they be described as homozygous dominant, homozygous recessive, or heterozygous? So, using Mendel's laws, we've sorted out the mystery as to why all of the members of the F1 generation were smooth. But we also know that Mendel went further and allowed these F1 generation plants to grow and mature and then self-fertilize. So, meaning the male gamete was allowed to pollinate the female gamete from the same plant. So let's look at what the genetic content of the gametes looks like in the F1 generation plants. So getting back to this graphic, these plants started out with germ cells that contain one of each allele. One chromosome has the dominant allele, and the other one has the recessive allele, because that's what they inherited from their true breeding parents. And then when these germ cells go through their cell cycle, they duplicate their DNA during S phase, and then the homologous chromosomes get separated in meiosis 1, and then finally the sister chromatids get pulled apart in meiosis 2. So, unlike with their parents, where 100% of their gametes had the same genetic content because they were true breeding, for the F1P, half of the gametes contain a chromosome that has the dominant allele, and the other half contain a chromosome that has the recessive allele. And this means that if you were to be able to peer into the ovules in the ovary and see what their genetic content was, exactly half of them would have a dominant allele, and the other half would have a recessive allele. And likewise, if you were to be able to peer into the pollen, half of those cells would have the dominant allele, and the other half would have the recessive allele. So, what happens when this flower self-pollinates? Well, with any fertilization, and this is true for all eukaryotes, including humans, there's a certain level of randomness at play. A lot of it has to do with which egg happens to bump into which sperm, or which pollen happens to bump into which ovule. So if you're looking at a situation where half of the gametes have the dominant allele and the other half have the recessive allele, there are 4 possible ways that these gametes could bump into each other. First, a pollen that contains the dominant allele, capital A, could bump into an ovule that also contains the dominant allele, capital A. You can see in the image on the right, I put a yellow box around an example of one of each of these. Second, a pollen containing the dominant allele could fertilize an ovule containing the recessive allele. Third, a pollen containing the recessive allele could fertilize an ovule containing the dominant allele. And then lastly, a pollen containing the recessive allele could fertilize an ovule that also contains the recessive allele. Now I'm going to let you take the reins from here in another checkpoint. In each of these 4 scenarios, what would be the genotype of the offspring produced from each fertilization? And then once you figured that out, determine what would be the phenotype of each of these offspring. And lastly, and this is what I really want to know as your ultimate answer to this checkpoint, how many of those possibilities would have smooth phenotypes and how many of them would have wrinkled phenotypes? Now, if you did that checkpoint correctly, then everything should finally come together in forming an understanding of Mendel's experiment and knowing why he found that the F1 generation peas were all smooth, and also why he found that the F2 generation showed this 3-to-1 ratio of smooth to wrinkled texture. And remember, to put this back in perspective, the reason why Mendel's work was so important is because the blending inheritance hypothesis was, at the time, the prevailing idea about how inheritance worked. His ideas were the first to hold water in explaining why blending inheritance often doesn't turn out to be true. So in summary, the conclusions that came out of Mendel's first series of experiments were these 2 ideas of the law of segregation and the law of dominance, which still form the foundation of our understanding of modern genetics well over a century later. But today, instead of going through the painstaking explanation of how each gamete is formed and the different possibilities for fertilizations like I just did, modern-day geneticists instead use a tool called a Punnett square that allows them to make predictions about what traits will be exhibited by the offspring of a cross. So Punnett squares are the streamlined tool for doing what we just did, using Mendel's laws to predict the genotypes and phenotypes of offspring. So let's see how this streamlined system works in the context of the F1 generation P. The first step in constructing a Punnett square is to determine the parent genotypes. In this case, we're working with the F1P, which, as we know, underwent self-fertilization. So really, this F1P represents both parents wrapped up into one. But of course, that's not always the case. But we know that the F1P plant's genotype was big A, little a. They had one of each allele. That's the parent's genotype. So step 1, complete. Step 2 is to determine what is in the parent's gametes, their reproductive cells. So what alleles do you find in there and in what proportion? And as we discussed during meiosis, the chromosomes will get separated, and half of the gametes end up with the dominant allele, and the other half end up with the recessive allele. And this is true for both parts of the plant. It's true for the pollen, and it's true for the ovules as well. Both have a 50-50 mix of dominant and recessive alleles. So there are 2 equally likely possible gametes for each parent. And this leads us to the third step, which is to create a table with a row for each of the male parent's gametes and a column for each of the female parent's gametes. So there are 2 genetic possibilities for the male parent, big A and little a, which means there should be 2 rows. And there are also 2 possibilities for the female parent, big A, little a, which means there should be 2 columns. So we set up a table that's 2 by 2. And if you take these gametes and you assign them to their own row and their own column, then the final step is to fill in the boxes in the middle of the table. So we do this by taking the allele for the male parent on the side and the allele for the female parent on the top, and you write them both in the box in the middle. So if we focus on the box in the upper left-hand corner, it gets 2 capital A's, one from each parent. And what you just did there, what that simulates is fertilization. You've shown one possibility, which is the possibility of a male gamete carrying capital A fertilizing a female gamete also carrying capital A. The 2 letters in the middle represent the genotype of the offspring. So to be clear, each box in this Punnett square represents a possible offspring. And we have to repeat that process for each of these boxes. So now looking at the upper right-hand corner box, This one gets a dominant allele from one parent and a recessive allele from the other parent. So this is another possible way that these gametes could fertilize. And note here that the conventional way of doing this is to write the capital letter first in the heterozygous pair. Doesn't matter which parent is male and which is female. Doesn't matter which one is the row or the column. The convention is you write the capital letter first. And then if we turn our attention to the lower left-hand box, this box also gets one capital letter and one lowercase letter. And then finally, the lower right-hand box, this offspring has the genotype lowercase a, lowercase a. Now, if we then take this Punnett square and we look at all four possible boxes in there and we figure out what the phenotype would be, we would conclude that 3 out of the 4 possibilities are smooth, and one of the 4 possibilities, specifically the lower right-hand one, is wrinkled. So in practice, this means that when two parents with the genotype big A, little a cross with each other, there is a 75% chance that the offspring will be smooth and a 25% chance they'll be wrinkled. And again, this agrees with what Mendel saw in his experiments. Now I want you to try a Punnett square on your own. To double check his hypothesis, Mendel went back and cross-pollinated an F1 generation pea with a true breeding wrinkled pea. And I'd like you to complete a Punnett square to predict the ratio of smooth to wrinkled offspring that you would expect to come from this cross. So here in the middle of the slide, I have the two parents labeled, with the male parent being the F1 pea and the female parent being a true breeding wrinkled pea. So go ahead and follow those 4 steps outlined on the previous slides to set up a Punnett square and tell me what you would expect the offspring of this cross to look like. And while I'm not going to tell you the answer to this question, I will tell you that when Mendel actually performed this test, he did get numbers that matched his expectation, that matched the Punnett square very closely. So that concludes our discussion of Mendel's first experiment. This is a great place to take a break if you're going to do so. And now we're going to move on to talk about one other really important reality that Mendel discovered about the inheritance of traits in a separate second series of experiments. So his second series of experiments were focused on observing different combinations of traits in pea plants rather than just a single trait. So for example, we just did a deep dive on the characteristic of texture, in which smooth is the dominant and wrinkled is the recessive trait. But how does texture interact with color? For seed color in pea plants, yellow is dominant and green is recessive. And we're going to continue to use the letter A to represent the alleles for texture, but now we're going to introduce the letter B to represent the alleles for color. So in one of these next level experiments, Mendel took a pea plant that was true breeding for the yellow phenotype and also true breeding for the smooth phenotype. That's the one on the left. And he crossed it with a separate plant that was true breeding for the green phenotype and true breeding for the wrinkled phenotype. And when he did this, he found that 100% of the F1 generation peas were yellow and smooth. They looked entirely like one parent and nothing like the other. Okay, so that's in line with what we saw before in the first experiments. There's nothing special there. But as you can probably guess, he didn't stop there. He let those F1 peas self-pollinate and produce an F2 generation. And what do you think that F2 generation looked like? If I ask you to guess based on what he saw in his first experiments, you might guess that he saw the green and wrinkled phenotype come back at a 3-to-1 ratio with the round and yellow peas. However, this is not what he saw. Instead, Mendel found that the F2 generation was made up of 4 different types of peas, and that the texture and color traits had mixed and matched with each other. So not only did this generation contain smooth yellow peas and wrinkled green peas like the parent generation did, but it also had smooth green peas and wrinkled yellow peas. And on top of that weirdness, the numbers that he got followed a very pattern. So he did these crosses for multiple combinations of traits, and he always found that the F2 generation the offspring of the F1 generation, the offspring that had both dominant traits, in other words, yellow and smooth, they ended up with 9/16 of the pool. And then the offspring that had one dominant combined with one recessive trait, like being smooth and green or being yellow and wrinkled, those each made up 3/16 of the pool. And then finally, the offspring that had both recessive traits together, green and wrinkled, made up the remaining 1/16. And he repeatedly saw these very specific numbers. And this led him to propose his last hypothesis for explaining this phenomenon. He proposed that when genes for separate traits are segregated into gametes, they do not travel as a unit. So this means that the inheritance of one trait does not guarantee the inheritance of another trait. Genes for separate traits, like color and texture, they're independent of each other. So that's how you end up with the traits mixing and matching in the F2 generation. Today, these explanations have been formalized under the title of Mendel's law of independent assortment. So let's look at why this is the case. Let's look at why traits are able to mix and match. For the yellow smooth parent, we know this parent was true breeding for the dominant version of both characteristics. So for their texture, both of their chromosomes contain the dominant allele for smoothness. And then on a separate chromosome, there's a gene for color, and both of those chromosomes contain the dominant allele for yellow color. So this parent's genotypes for these two traits is capital A, and then capital B, capital B. And then on the other hand, the true breeding green wrinkled P is homozygous recessive for both traits. So their genotype is going to be lowercase a for the wrinkled texture, and then lowercase b for the green color. So let's take a look at what happens when the smooth yellow parent produces its gametes. So here we have a germ cell in the smooth yellow parent. You can see its genotype is labeled on the chromosomes. During S phase of the cell cycle, these chromosomes get duplicated, and then in meiosis 1, the homologous chromosomes are pulled apart into two separate cells, and then when meiosis 2 comes along, the sister chromatids split, and you get haploid gametes that have one allele for each characteristic. And that's an important point to note here, because do you see how each of these gametes has an A and also a B? There's one allele for each characteristic. And of course, all of the gametes are identical because this parent was true breeding. So it's inevitable that this parent passes down the dominant allele for both characteristics. And I'll spare you the tedium of going through the visuals for the other parent. I trust that by now you understand that the true breeding green wrinkled P would end up in the same situation, except all its gametes contain the recessive alleles. Consequently, when you bring these two parents together, when the pollen cross-pollinates with the ovule, all of the offspring are going to receive the dominant alleles for both traits from one parent and the recessive allele for both traits from the other parent. So the fertilized embryos will all have the genotype capital A, lowercase a, capital B, lowercase b. The pair of A's will cause the offspring to be smooth in texture because the dominant overrides the recessive. And then the pair of B's will cause the offspring to be yellow in color because, again, the dominant overrides the recessive. And as a result, all of those F1s are yellow and smooth. Now that conclusion was pretty obvious. That's the easy part. When we go from the F1 generation to the F2 generation is where things start to get a little wild. And you'll see why when we look at how the gametes are formed in the F1 pea plants. So here we have the germ cell in the yellow smooth parent. You can see its genotype labeled on the chromosomes again. During S phase of the cell cycle, those chromosomes get duplicated. They each sprout a second pair of identical arms. And then in meiosis 1, one of the chromosomes gets segregated into one cell. The other one gets segregated into the other cell. And if by now you're sort of tuning out these little walks we're doing through meiosis, at least pay attention to this point right here. Each cell that comes out of meiosis 1 is going to get a chromosome with the gene for texture. It's going to get an A chromosome, and it's going to get a chromosome with the gene for color, a B chromosome. So it gets an A and a B. But the way that they segregate will be random. In this illustration, what you see is that the chromosomes with the dominant alleles ended up in the same cell. That's the top cell. And the chromosomes with the recessive alleles ended up together in the bottom cell. And in this situation, when these cells go through meiosis II and the sister chromatids get separated, you'll end up with two gametes, the top ones, that contain both capital letters alongside two gametes that contain both recessive letters, which is the bottom ones. But if we take one step backwards, it's just as likely that these chromosomes could have segregated differently in meiosis I because it's entirely random which chromosome moves to which side of the cell. So there's just as good of a chance that the recessive chromosome for texture could have ended up in the same cell with the dominant chromosome for color. So that's the situation you see on the top now. And then on the bottom, we have the dominant texture alongside the recessive color. And if this is the case, then when these cells undergo meiosis II, the sister chromatids will separate and you end up with a set of gametes. They're still haploid, but they have a different set of alleles in them. So now we've got a little a with a big B for the top two and a big A with a little b for the bottom two. So to summarize, the F1P is going to give us 4 genetically unique gametes. Some gametes will contain the dominant alleles for both traits. Some will contain the recessive alleles for both traits. Some will contain the recessive allele for texture alongside the dominant allele for color. And then lastly, some will contain the dominant allele for texture alongside the recessive allele for color. And each one of these occurs in equal proportions. A quarter of the gametes will be made up of each of these combinations. So if we had the ability to peer into those reproductive cells within the pea plant, we would see that the ovules in the ovary are equally distributed among those 4 possibilities, and that the male gametes in the pollen are as well. And again, when this flower self-pollinates, randomness will dictate which pollen bumps into which ovule. But now that we're considering 2 traits at the same time, there's going to be actually 16 different possible ways that these gametes could combine. Now, I'm not going to make you suffer through an exhaustive look at all 16 of them, but just to give you a sense of the complexity that we're dealing with, a pollen containing both dominant alleles could fertilize an ovule containing both dominant alleles. A pollen with both dominant alleles could fertilize an ovule with the recessive wrinkled alongside the dominant yellow. And then a pollen with both dominant alleles could fertilize an ovule with the dominant smooth allele and then the recessive green allele. And then pollen with both dominant alleles could fertilize an ovule with both recessive alleles, and so on and so on. Ultimately, there's 16 different possibilities. Obviously, this is a tedious business, but again, we can streamline it by bringing back that trusty tool that we just learned about, the Punnett square. Because Punnett squares don't just work for predicting the outcome of the cross of a single characteristic, they also work for multiple characteristics. And they work in the exact same way. So let's see it in action by using it to predict the outcome of this F1 self-fertilization. The first step is, again, to determine the parent's genotypes. We know that the F1P was heterozygous for both of the characteristics under consideration. It had 1 capital letter and 1 lowercase letter for each of the traits. The second step is, again, to determine what is in the parent's gametes. And in this case, what allele pairs could possibly be found in there. So as we just saw, the chromosomes are going to sort themselves into the gametes independently of each other. They can mix and match, so therefore there's 4 possible genetically unique gametes. So we've got capital A, capital B, lowercase a, capital B, capital A, lowercase b, and lowercase a, lowercase b. And because this is a self-fertilization, the same parent is supplying the female gametes as well, so we have the exact same range of possibilities for the other parent. So each parent has 4 gametes for these 2 traits. The third step is to create a table with a row and a column for each one. So because there's 4 possibilities for each parent, we make a table that is 4 rows by 4 columns, and then we assign one of these possibilities to each of the rows or each of the columns. The final step is to fill in the boxes in the middle, which, remember, each box represents an offspring. So in that first box in the upper left-hand corner, first we bring down the capital A from the top and the capital A from the side. And then we bring the capital B from the top and the capital B from the side. And note that the letters of the same type are paired together. So what we did is we first gathered the A's and we paired them together. And then we gathered the B's and we paired them together. And we repeat this process for each of the boxes. For the box below the one that we just did, we bring down the capital A from the top and the lowercase a from the side, and then the 2 capital B's. And then for the box below that one, we bring down the capital A's, and then the capital B from the top and the lowercase b from the side. And now note that when we did this, we still put the capital letter first in the heterozygous pairs. And then for this last box in that same column, we bring down the A from the top and the A from the side, B from the top and B from the side. And now that first column is complete. And now I'm just going to go ahead and jump ahead and fill in the rest of the boxes for you in one swoop here. And now what this represents is all of the different possible genotypes of offspring that can come from this cross. But what do they look like? We know their genotypes, but what are their phenotypes? To find this out, you have to look at each of the individual boxes like we did before and determine what traits will be expressed based on the letters that the offspring has. So the pea plant represented by that very first upper left-hand box will be smooth and yellow. So will the one below that. So will the one below that. And so will the one at the bottom. That entire first column is smooth and yellow because each one of those offspring has at least one capital A, making it smooth, and at least one capital B, making it yellow. And we can go box by box through that table, and eventually you would find this. So each of these has been color and texture coded for what the offspring would look like. And if you tally up how many representatives there are of each phenotype, what you would find is that 9 out of the 16 boxes are yellow and smooth, 3 out of the 16 are green and smooth, 3 are yellow and wrinkled, and only 1 is green and wrinkled. And this agrees beautifully with what Mendel found in his numbers. He found those same ratios on a larger scale when he did this with hundreds of pea plants. And these are the experiments that gave him the support for his explanation about how chromosomes sort, which is what we now know as the law of independent assortment. Genes for separate traits do not travel as a unit, and inheriting one trait does not guarantee the inheritance of another. They mix and match. But there's one lingering question that many students have about this idea, which is the following. When I showed you how these genes for texture and color sort independently of each other, we had them placed on 2 separate chromosomes. But, as we know, chromosomes contain multiple genes. So what if you're studying 2 genes that are both located on the same chromosome, just different sections of that chromosome? Won't those genes always travel together into the same gamete, and then the law of independent assortment wouldn't hold true? And if that question jumped to your mind, it's a good thought and it's very logical. However, in these cases, independent assortment does still hold true. And the reason why is because of the phenomenon that we learned about in our last lecture called crossing over. So remember, crossing over is this thing that happens during meiosis where segments of homologous chromosomes are physically swapped with each other. And because this happens during gamete formation, you will have a random assortment of genes that get swapped between chromosomes. It could be the color gene, it could be the texture gene, it could be both. but it happens so many times that you still end up with these genes behaving as if they were independent of each other, even if they're located on the same chromosome and would be expected to travel together. So I now have another checkpoint for you, which is similar to the checkpoint that we did when learning about Mendel's first two laws. So just like in his first series of experiments, to double check his hypotheses, Mendel cross-pollinated an F1 generation plant with a true breeding wrinkled plant. We're going to simulate that cross in a 2-trait Punnett square and see what the offspring were expected to look like. But I don't want you to go all the way in this checkpoint. We're going to break this up into 2 parts. So for the first part here, I want you to complete just the first 2 steps, which are, what are the parents' genotypes? And then once you know the parents' genotypes, tell me what pairs of alleles could each parent have in their gametes. So please use the same letters we've been using. Use A for texture and B for color, and list all the possibilities for each parent. And remember, when you're assembling those pairs, keep in mind that every gamete should contain an allele for color alongside an allele for texture. So if you end up in part 2 of this question saying that there's gametes that have 2 A's together or 2 B's together, that's not correct. And then once you have this information, you have what you need to set up your Punnett square. So for checkpoint 8, that's what I'd like you to do. Create the Punnett square with the right number of rows and columns based on what you just concluded about the number of possible allele pairs. So you'll use your answer from checkpoint 7 to figure out the rows and columns in checkpoint 8. And then you'll fill in the boxes to show the offspring in the middle. And I'll give you a hint, this Punnett square does not look like the one that we walked through together that had 16 boxes. It's much smaller than that giant Punnett square that we just saw. And ultimately, your goal here is to give me a prediction about the percentage of offspring that have each phenotype combination. So what percentage are yellow and smooth? What percentage are green and smooth, yellow and wrinkled, and green and wrinkled? And again, I won't give away the numbers because that would tell you the answer, but I will tell you again that when Mendel did this cross, he got percentages that agreed very closely with the Punnett square prediction, which then validated his hypothesis about independent assortment. So we're getting very close to the end of our journey talking about genetics, but we can't just leave things there with Mendel's experiments and laws because that's not the whole picture. Do you remember how we started the lecture talking about the blending inheritance hypothesis? Well, there's a reason why people thought that offspring inherit the average of their 2 parents. Because it's something that really happens, like with human skin color, for example. And this doesn't agree with Mendel's laws. So what's going on here? The truth is that Mendel offered us a good start in our understanding of genetics, but in reality, very few genes and traits work as simply as the ones that Mendel studied in his Ps. For example, it's very rare for a characteristic to be determined by only one gene. In Mendel's Ps, there was a single gene that controlled texture and a single gene that controlled color. Human skin color, on the other hand, is controlled by over 150 different genes that all contribute to the final picture. This is known as a polygenic trait. The prefix poly means multiple, and a polygenic trait is one that's controlled by more than one gene. It's also very rare for a characteristic to only have 2 different alternatives. In Mendel's Ps, there were 2 options for each characteristic. There was green and yellow. There's wrinkled and smooth. But that's usually not the case. For example, there are way more than just 2 human eye colors. And this describes a situation referred to as multiple alleles, where there's more than 2 gene variations, which complicates things beyond Mendel's laws. It's also very rare for one allele to be completely dominant and another to be completely recessive. Sometimes the dominant allele does not completely block out the recessive allele. For example, in snapdragons, which are a type of flower, the allele for red flowers only partially masks the allele for white flowers, which means that heterozygous plants, ones that have one dominant and one recessive allele together, they end up expressing a blend of red and white, and therefore they are pink. This situation is known as incomplete dominance, and this also applies to human skin color. Human skin color is both a polygenic trait and a trait where incomplete dominance is at play. Now let's stop briefly here and tackle another checkpoint. I'd like you to complete a Punnett square to cross two heterozygous snapdragon plants. And you can use any letter you'd like for this. What percentage of their offspring would you expect to be red, pink, and white? There are a few other variations in inheritance patterns that are worth mentioning before we close this lecture. One of these is gene-environment interactions. Gene-environment interactions are where a combination of a person's genes and their environmental exposures produce a trait. So the genes themselves alone are not enough to bring about the phenotype. There has to be something that happens in a person's lifetime to trigger the effect. One example is the disease multiple sclerosis, which is an autoimmune disease where a person's immune system attacks the cells in their nervous system and causes problems with muscle control. The cause of this disease is not fully understood, but it's known that a combination of genes and environmental factors play a role. And we know this because related people who are related to each other, they're more likely to be affected, meaning that there is some genetic component that runs in families. But those genes are not enough by themselves. I once taught a student who was an identical twin, meaning she shared 100% of her DNA with her sister. My student had multiple sclerosis, and her twin sister did not, even though they were completely genetically identical. So something more has to be at play. And various studies have suggested that environmental exposures like smoking, childhood obesity, and infection with a virus known as mononucleosis or mono are what trigger the development of the disease in people who have these certain genes. Another inheritance variation is sex-linked traits. Sex-linked traits are caused by genes carried on the X chromosome or the Y chromosome. The expression of these traits varies depending upon which set of chromosomes a person has. So an example of a sex-linked trait is the gene for proteins that are involved in blood clotting, which are carried on the X chromosome. Mutations in these genes can cause a recessive disease known as hemophilia. When a person suffers from hemophilia, small bumps and cuts can be life-threatening because their blood lacks the ability to clot. And so minor injuries can lead to serious blood loss. And thankfully, this disease is recessive, but males are more susceptible to it because of the set of chromosomes they have. For females, and I use these terms male and female loosely because, as we just learned in Chapter 7, there is more to biological sex than just the set of chromosomes. But for females, because they have two X chromosomes, there are three possible genotypes. They can have the healthy dominant allele on both of their X chromosomes, and this is how sex-linked traits are written. You can see there's the sex chromosomes that are the base, and then the allele is a little superscript, that little capital H represents the healthy allele. And if a female has two copies of the dominant allele, like you see here, of course they're healthy. They don't have hemophilia. A female can also have one copy of the dominant allele on her first chromosome, but be the carrier of the recessive allele for hemophilia on her second X chromosome. But a person with this genotype still doesn't have hemophilia because the dominant allele masks the disease. Only if a female has two copies of the recessive allele on both of her X chromosomes will she suffer from hemophilia. Now, on the other hand, for males who have just one X chromosome alongside one Y chromosome, if they have the dominant healthy allele on their one X chromosome, of course they are healthy. And if they have the recessive allele on their one X chromosome, then they have the disease. For males, it's not possible to be a healthy carrier of the disease. It's not possible to be heterozygous because they don't have a second X chromosome. And the Y chromosome has nothing to do with blood clotting. The genes for blood clotting simply aren't there on the Y chromosome. So males are stuck with whatever they get on their X chromosome. And that's why for sex-linked traits that are carried on X chromosomes, males are more susceptible to disease states. A very visible place in history where this pattern has played out is in the lineages of some of the royal families of Europe. So Queen Victoria of England, who I just boxed in green, This is the person for whom the Victorian era is named. She was a carrier of the disease hemophilia. She didn't suffer from it herself, but she was heterozygous and she was a carrier. And she had 9 children, and she passed the allele for hemophilia down to 3 of them. 2 of them were daughters, and 1 was a son. So in this graphic, women are represented by circles, and men are represented by squares. And you can see there's a key in the little box toward the bottom where you can see what every symbol means. So as you can see, both of Queen Victoria's daughters, the circles, they were just carriers because they got a healthy X chromosome from their father, which masked the hemophilia. And then the son, Leopold, received a Y chromosome from his father instead of an X chromosome. So he had no chance to mask the disease, and therefore he had hemophilia. And you can see how as these lineages continued, her children continued to pass down the allele, but the daughters, the circles, they were always just carriers, while it was the sons, the squares, who were actually affected by the disease. So this shows a real-life case of how the inheritance of sex-linked traits plays out differently in men versus women. And of course, we can't wrap things up without quickly explaining what's going on with the Labradors and their coat colors. Labrador coat color is dictated by a pattern known as epistasis. This is where one gene blocks the expression of an entirely separate gene. So labs have 2 genes that play into coat color. One is called the MC1R gene, which determines whether the skin cells produce a pigment that is carried into their fur. For this gene, pigment production is dominant, and I've put a capital E there to represent it, and a lack of pigment production is recessive, so lowercase e. But then there is a separate gene called TYPR1, which determines what color pigment the skin produces if they produce one. And for this gene, black pigment is dominant, so capital B, and brown pigment is recessive, lowercase b. So yellow labs do not produce a black or brown pigment. That's because they have a homozygous recessive genotype for their MC1R gene, little e, which tells their cells not to make pigment. They still have a TYPR1 gene in their chromosomes, but its expression is being suppressed by their MC1R gene. So I like to say that every yellow lab is secretly a chocolate lab or a black lab, and we just don't know it. Their genotype could be any of the 3 possible genotypes for pigment color. It could be capital B, it could be capital B, lowercase b, or it could be lowercase b. And you would never know unless you spent thousands of dollars to get your dog's DNA sequence to find out, because the 2 little e's are masking the expression of the other gene. So for our final checkpoint of this lecture, we're going to turn our attention back to the case of the 2 labs who produced a mixed litter of black, brown, and yellow puppies. And I'd like you to tell me what the parents' genotypes must have been for this to be the case. Remember, the parents were the 2 black labs, but they produced all 3 different possible colors of puppies. And remember, there's 2 traits here. One of them is, do the cells make a pigment? And what color is the pigment is the second trait. So for each parent, you'll need to specify 4 letters. 2 of them are E's, 2 of them are B's. What must the parents' genotypes have been to produce this litter? And I'll give you a clue. Both parents have the same genotype. A good way to approach this problem is to think about what the puppy's genotypes would have to be, and then sort of reverse engineer it to figure out what the parents must have had. Now, after hearing about all of these complexities, you may be left wondering, why do we even care about Mendel's laws? It seems like they don't really apply in most cases. And yes, it is true that most genes and traits operate at a level of complexity that's far beyond what Mendel was observing. However, Mendel's work is still important, and it's because Mendel studied simple traits that he was able to see past all of the complexity and come up with the correct mechanism of inheritance that other people hadn't been able to see before, which is that characteristics are brought about by a pair of genes. One gene is inherited from the male parent and the other from the female parent. The parents' genes get separated and randomly distributed into their gametes. And the genes for different characteristics are independent of each other and therefore can be inherited in any combination. So these lessons from Mendel's work form the foundation of our understanding of genetics today.